Hello everyone. In this lecture, we are going to study some of the basics of hemoglobin, and I will discuss only the important points that we should definitely know before studying the hemoglobinopathies. So, hemoglobin is a combination of heme plus globin. You must be knowing that heme synthesis happens in the mitochondria, and there are some steps in the heme synthetic pathway which happens in the cytoplasm also. And the predominant site of heme synthesis is the bone marrow. Okay. Now, if you see the heme synthetic pathway, again, I will not go into the details of the pathway because it's slightly complex and it is not important at this point of time to understand all the steps. We'll discuss that when we'll study about the porphyrias. So, if you see the pathway, it starts from the reaction between succinyl-CoA and the glycine. The first step is the reaction between succinyl-CoA and glycine, and this reaction is catalyzed by the enzyme ALAS, that is ALA synthase. ALA is aminolevulinic acid. Okay, aminolevulinic acid. And this enzyme can cause, it will catalyze the reaction between succinyl-CoA and glycine, and it will produce aminolevulinic acid. So that is the first intermediate product in the process of heme synthesis. Now, this ALA synthase requires a cofactor that is the pyridoxine or vitamin B6. Now, the second step is catalyzed by ALA dehydrogenase, and this ALA dehydrogenase will convert the aminolevulinic acid into porphobilinogen. Now, there are various steps inside the cytoplasm which will ultimately convert the porphobilinogen into protoporphyrin 9 inside the mitochondria. So, the protoporphyrin 9 will be formed inside the mitochondria. And in the last step of heme synthesis, the protoporphyrin 9 will combine with iron molecule with the help of ferrochelatase to form the heme. Okay, so heme is synthesized inside the mitochondria and the globin is synthesized in the cytoplasm, and they will together form the hemoglobin molecule. Now let us see what is going to happen if the patient is having vitamin B6 deficiency. So if there is pyridoxine deficiency, this process, the first step cannot happen. So there is a defect in the heme synthetic pathway. So this will lead to decreased production of the protoporphyrin 9. And if there are not enough protoporphyrin 9 to attach to the iron molecule, there will be decreased heme synthesis. So that will cause anemia. Okay. So pyridoxine deficiency can ultimately cause anemia because of reduction in heme synthesis. Reduction in heme synthesis happens because of reduction in production of protoporphyrin 9. Okay. So that will ultimately cause anemia. But one more thing, you have to appreciate that if you do not have protoporphyrin 9, what this iron molecule is going to do? Now, this iron molecule will accumulate inside the mitochondria. Now, this abnormal accumulation of iron inside the mitochondria is known as the sideroblast. Okay, so if RBC is having numerous iron particles abnormally deposited inside the mitochondria, now this kind of RBC is known as the sideroblast. So this B6 deficiency or pyridoxine deficiency will cause sideroblastic anemia. It will cause sideroblastic anemia. Similarly, in lead poisoning, basically this lead can actually inhibit the ferrochelatase. It can inhibit the ferrochelatase. Also, lead can inhibit. It can also inhibit the ALA dehydrogenase. It can also inhibit this enzyme. So, because of inhibition of these enzymes, ultimately heme cannot be synthesized, and this iron, the free iron, will tend to accumulate inside the mitochondria, forming a sideroblast. So, lead poisoning will also cause sideroblastic anemia, and pyridoxine deficiency will also cause sideroblastic anemia. There are various other causes of sideroblastic anemia also. We'll discuss that when we'll discuss about the sideroblastic anemias. Okay. Now, as you understood this, now we'll see what is the structure of heme. So, heme is nothing but, as we discussed, it is protoporphyrin 9 plus iron. Protoporphyrin 9 is actually a tetrapyrrole molecule. As you can see here, these are pyrrole molecules. These are pyrrole rings, and combination of 4 pyrrole rings is known as a tetrapyrrole molecule, and this protoporphyrin 9 is a tetrapyrrole molecule. And along with iron, they will make this heme. Now, with this heme, a globin chain can be attached, and globin chain will be attached to the iron molecule. It will be attached to the iron molecule. So if you combine this heme plus globin, this is the globin chain. If you combine the heme with globin, they will make hemoglobin. But this is a hemoglobin monomer. This is a hemoglobin monomer. The hemoglobin that we find inside our body, they are in tetrameric structures. Okay, so these are tetramers. Tetramers means there are 4 hemoglobin monomers. Okay, so these are composed of 4 hemoglobin monomers. Now these hemoglobins are named based on the globin chain they have. Okay, so in each tetramer, usually you are going to get 2 types of globin molecules. 2 types of globin molecules. For example, in adults, the most common hemoglobin is hemoglobin A. So it is composed of alpha 2, beta 2, alpha 2, beta 2. That means we will have 2 alpha globin chains and 2 beta globin chains. And usually these tetramers are actually a combination of heterodimers. What do you mean by heterodimer? That means, as you can see here, if this is a alpha molecule, this is alpha globin chain, this is a beta globin chain. Now they will make a dimer that is heterodimer. It is because it is composed of different globin chains. So this will be a single heterodimer. Similarly, here also they can produce another heterodimer. And if these heterodimers they combine with some bonds, they will make the tetramer. They will make the tetramer. And in adults, most common hemoglobin A, that is alpha 2, beta 2. It is composed of 2 dimers of alpha beta. Okay, so alpha beta, 1 heterodimer. They will form bonds with another alpha beta heterodimer, and they will ultimately cause this form this alpha 2 beta 2. Okay. So as you understood this, now we'll go into the globins. But before going into globin, I'll just recap the oxygen dissociation curve again. This we have also discussed when we discussed about the properties of mature RBCs. I want to discuss it again because this is very important to understand some of the pathophysiology of hemoglobinopathies, especially the sickle cell disease. Now, we have discussed that the normal hemoglobin dissociation, oxygen dissociation curve is sigmoid shape. And this sigmoid shape is because of the phenomenon of cooperativity. So basically hemoglobin, as I told, it's a tetramer. So I'll just do a schematic diagram. So this is a tetramer. And this tetramer is composed of 4 monomers of 4 hemoglobin monomers. And each monomer has 1 iron molecule. Each monomer has 1 iron molecule. Now each iron molecule can bind to 1 molecule of oxygen. So they can bind to 1 molecule of oxygen. Now what you need to understand is the hemoglobin, the normal hemoglobin, which is a tetramer, it will not, all the iron molecules will not bind to oxygen simultaneously. So what will happen? Whenever you add 1 molecule of oxygen to 1 iron molecule, the other monomers will develop increased affinity for further oxygen binding. Okay, so this is known as the cooperativity. That means I'll tell it again. When 1 monomer will bind to oxygen, the oxygen affinity of other monomers will increase. Similarly, when you take out 1 oxygen from one of the monomers, the other molecules also will reduce their oxygen affinity. Okay, so if you give 1 oxygen, it is easy to oxygenate the other monomers, and if you remove 1 oxygen, it is easy to remove other oxygens from other monomers. Okay, so this is known as the phenomenon of cooperativity. Now, how this happens, that we can understand from this diagram. So, if you take out 1 oxygen molecule from the hemoglobin, there will be formation of some salt bonds. These salt bonds will make the T configuration, which is otherwise known as the TENS configuration, or sometimes known as the taut configuration, whatever you want, you can tell. So basically, if you take out 1 oxygen molecule, they will make some salt bonds. Once the salt bonds are formed, the hemoglobin will take a TENS form, TENS configuration. Okay, now this TENS configuration will have reduced oxygen affinity. It will have a reduced oxygen affinity. Okay, so that means if you take out 1 oxygen, they will tend to lose the other oxygens. Similarly, what will happen whenever you add oxygen? So if you add oxygen, these salt bonds will be broken. These salt bonds will be broken. And if you break the salt bonds, they will make a R configuration, which is otherwise known as the relaxed configuration. relaxed or R configuration. Now, if the hemoglobin molecule is relaxed, it can take up more and more oxygen. It can take up more and more oxygen. Okay, so I'll repeat it again. So if you have a hemoglobin molecule which is a tetramer which can take up 4 molecules of oxygen, if you add one oxygen, the salt bonds will be broken, they will become more relaxed, and they will tend to take up more and more oxygen. That means the affinity for the oxygen will increase. Why it is increased? Because they are relaxed. Why they relaxed? Because they have no salt bonds. But if you take out one oxygen molecule, there will be formation of salt bonds that will make the hemoglobin to take up the tensed configuration or taut configuration, and this configuration has reduced affinity for oxygen, and then they will tend to lose further oxygen. They'll tend to lose the oxygen. Okay. So that is why they make this sigmoid curve. They'll make this sigmoid curve. This is because of the phenomenon of cooperativity. Okay. So the sigmoid curve, basically it has a steep portion, steep area in the mid range of oxygen. So basically here the x-axis is nothing but the partial pressure of oxygen. This we have discussed previously, and the y-axis is the oxygen saturation of the hemoglobins. Oxygen saturation. So you can understand that if you make a small increase in the partial pressure of oxygen, there will be a larger increase. There will be a larger increase in the oxygen saturation. This is because of the cooperativity. Okay, so that is why the curve is sigmoid shape. Okay. Now there are some factors we'll discuss. There are some factors which will shift the curve towards right, and there are some factors which will shift the curve towards left. Okay, so this also we have discussed previously. Okay, before going into the causes, what is P50? So what is the concept of P50? P50 is the partial pressure of oxygen at which hemoglobin is 50% saturated, and normally the P50 is 27 millimeter of mercury. Okay, so at 27 millimeter of mercury, the hemoglobin is 50% saturated. So that is the P50. Okay, now we'll see what are the factors which will cause a rightward shift. Rightward shifting means what? Rightward shifting means the oxygen affinity will reduce and the hemoglobin will tend to release the oxygen. Okay, so rightward shifting means oxygen affinity is reduced. That means they will become, they will form more tense configuration, and so that they can release the oxygen. And leftward shifting means they will become more relaxed and they will increase their oxygen affinity. Leftward shifting means oxygen affinity is increased and they will tend to hold the oxygen rather than releasing the oxygen. Okay, so now let us discuss the causes. I would say let us recollect what are the causes that we have studied previously. Now we have studied that whenever there is increased CO2 or reduced pH means it is similar to increased H plus. I mean, increased acids will reduce the pH. So these conditions will cause a rightward shifting, and these conditions happen at the tissue level. At the tissue level, there will be production of carbon dioxide, and there will be, because of some metabolic processes, there will be production of acids, especially the lactic acid. Now, in those conditions, the tissue will need more and more oxygen. So that is why in these conditions, there will be rightward shift that will cause release of oxygen from the hemoglobins. If you want to understand the mechanism, so basically when the carbon dioxide will go into the RBCs, it will, along with water molecule, they will form this H2CO3. This H2CO3 will dissociate into HCO3, bicarbonate, and the proton molecule, H plus. Actually, this H plus, when it binds to hemoglobin, this H plus, when it will go and bind to the hemoglobin, it will actually convert this R form to T form. That means the H plus molecule can actually make the relaxed form into a taut form, and taut form has reduced oxygen affinity. So that is how it will release the oxygen. So that is how it will cause a rightward shift. Similarly, if there is a decreased pH, mostly because of lactic acid, which is a product of anaerobic metabolism. So decreased pH is associated with increased H plus, and that can directly bind to hemoglobin, and it can convert the relaxed form to the taut form, and it will cause rightward shift, and it will cause release of oxygen to the tissues. Similarly, whenever there is increased temperatures, and one molecule, a very special molecule, that is 2,3-BPG, 2,3-BPG, 2,3-bisphosphoglycerate. This also we have discussed previously. Now this molecule can actually shift the carb towards right and release the oxygen. Similarly, there are certain other factors like low oxygen affinity hemoglobin molecules, low oxygen affinity hemoglobin variants. So there are certain variants of hemoglobin which will have a inherent property of low oxygen affinity that can also cause shifting of carb towards the right. Left shifting, the exactly opposite things. So whenever there is reduced CO2, reduced H plus or alkaline pH, in all these conditions, there will be leftward shifting of the carb. That means the oxygen affinity will increase and they will tend to hold the oxygen rather than releasing it. Now this, I forgot to tell. So this phenomenon, whenever there is increased carbon dioxide, there will be rightward shifting and release of oxygen from the hemoglobin. Now this phenomenon is known as Bohr's effect. Bohr effect. This you must have studied in respiratory physiology also. So this is what happens at the tissue level. At tissue level, when they produce carbon dioxide, this carbon dioxide will go into the RBCs. They will cause release of oxygen from the hemoglobins. This is what happens at the tissue level, and this is known as the Bohr effect. The exact opposite effect happens in the pulmonary circulation. When the oxygen concentration is high, in pulmonary circulation, because of high oxygen concentration, there will be release of carbon dioxide from the hemoglobin to the alveolus. So that is what happens in the lungs. So that phenomenon is known as Halden effect. So Halden effect is opposite of Bohr effect. Bohr effect says increased carbon dioxide will reduce the oxygen affinity of hemoglobin, and Halden effect is increased oxygen concentration will reduce the carbon dioxide, will reduce the affinity of carbon dioxide with the hemoglobins. Okay, so that is what is Halden effect. Okay, so what are the other causes of left shifting? We'll study about hemoglobin F. So hemoglobin F is a fetal hemoglobin, which is basically alpha 2 and gamma 2. I told you adult hemoglobin is alpha 2, beta 2. Hemoglobin F is alpha 2, gamma 2. Now this also has increased affinity for the oxygen. So that will also cause leftward shifting. Now hemoglobin F, otherwise known as the fetal hemoglobin, this is found in the fetus. Now why it is important? Because fetus hemoglobin has to take up the oxygen from the maternal blood and supply to the fetus. So that is why they have high oxygen affinity. Because they have high oxygen affinity, they can take oxygen from the maternal circulation and they can provide it to the fetus. So that is why fetal hemoglobin is required for us, for the fetus. And also there are some certain high oxygen affinity variants that can also cause leftward shifting of the hemoglobin, oxyhemoglobin dissociation curve. So this is the concept of ODC, and these concepts are important to understand various pathophysiology that we are going to discuss when we will discuss about the hemoglobinopathies. Okay, in the previous slide I have shown you something like this, beta 4 and gamma 4. So what is beta 4 and gamma 4? Beta 4 means this is actually a type of hemoglobin. Here actually this beta 4 tetramer is containing 4 globin monomers, 4 beta globin monomers. I have told you previously that usually the hemoglobin tetramers are combination of heterodimers. That means you need at least 2 types of globin chains. I've discussed that for adults you need alpha, 2 beta. Similarly for fetus also I told you need 2 alpha, 2 gamma. But what happens in patients who have alpha thalassemia? That means the alpha globin is not produced. In those conditions, the beta chains, they tend to make tetramers, and in severe conditions, the gamma chains can form tetramers. But these hemoglobins are not desirable. They are unstable. They are not desirable. They will have various adverse effects. We'll discuss that in detail when we'll discuss about the alpha thalassemias. So these hemoglobins, this beta 4 and gamma 4, both of them will have increased oxygen affinity. And this will also cause leftward shifting of the oxygen dissociation power. Okay, so this point is clear. Now we'll study about the globin chains. What are the various globin chains and what are the various hemoglobins that we have? Okay. So we'll start with the globin gene anatomy and features. First, we'll understand what are the globin genes. So there are two chromosomes that we have to remember. Chromosome 11, which will contain the beta globin gene cluster, and and the chromosome 16 will contain alpha globin gene cluster. Okay, so chromosome 11 will contain beta globin gene cluster. So what is beta globin gene cluster? As you can see here, there is a gene here which is producing epsilon globin. Similarly, this gene is producing G gamma globin, and this is producing A gamma globin. Similarly, this is producing the delta globin. This is producing the beta globin. So all these epsilon, gamma, delta, beta. I'm repeating it again. Epsilon, gamma, delta, beta. Epsilon, gamma, delta, beta. These all comes under beta globin gene cluster. Beta globin gene clusters. So epsilon, gamma, and delta, these are also known as the beta-like genes. I mean beta-like globins. Okay, so I'm repeating it again. Epsilon, gamma, delta, beta. They all comes under this beta globin gene cluster. And epsilon, gamma, and delta, these are otherwise known as the beta-like genes. Similarly, if you see chromosome 16, it will contain the alpha globin gene cluster. So this will have genes for zeta globin, and it will have also genes for alpha globins. For alpha globin, we have two genes, and for gamma globin also we have two genes. But what is important to remember is this alpha globins. Alpha globins have two genes. Okay. The importance of this we'll discuss when we'll discuss about the alpha thesis. Okay. So this is what we have understood. So beta globin like structures beta like globins include epsilon, gamma, and delta. Similarly, alpha like globin include the zeta globin. Okay. Now, whenever a hemoglobin is formed, it should contain two globin chains from the alpha globin gene cluster and two globin chains from the beta globin gene cluster. Okay, as you can see here, alpha with beta can form alpha beta, alpha 2 beta 2, that is the adult hemoglobin. Alpha with gamma can produce alpha 2 gamma 2, that is the fetal hemoglobin. Similarly, zeta can also form some hemoglobin along with epsilon. So that will be something like epsilon 2, zeta 2, epsilon 2. Okay, so they can also make some hemoglobins, but I'm not going to discuss about that because those are embryonic hemoglobins. Okay, those are embryonic hemoglobins, and these are found only in embryos, and it's not important for us to remember. Okay, beyond this, beyond the epsilon and zeta, we have to remember and we have to understand everything. Okay, so what I have written here is what globin genes will be produced. But if you want to know the name of the genes, so basically the two genes for alpha globin chain is HbA2 and HbA1. Similarly, for gamma globin, we have HbG2, HbG1. For delta, the gene is HbD, and for beta, the gene is HbB. Okay, so these are the name of the genes. Okay, so HbG2 gene will produce the G gamma globin. HbG1 gene, this will produce A gamma globin. Similarly, HbD gene will produce the delta globin. So this is the HbD gene. Similarly, HbB gene will produce the beta globin. Okay, so like this. So these are the name of the genes, and the globin genes that are produced I have already written. Now the next point you have to remember is what is the predominant beta-like globin before birth and what is the predominant beta-like globin after birth. So predominant before birth and predominant after birth. So before birth, the predominant beta-like globin is gamma, and predominant after birth is beta, and to some extent delta. Beta more than delta. I hope it is clear. So before birth, the predominant beta-like globin that is produced is the gamma globin. And among the alpha gene and after birth it is the beta globin chain. And among the alpha gene clusters, both before and after birth, the alpha globin chain is predominant. Zeta is only found during the embryonic period. Okay, so I hope it is clear. So before birth, the predominant beta-like globin is gamma, and after birth, it is the beta globin chain that is predominant. But in among the alpha-like genes, alpha-like globins, it is the alpha globin that is the predominant form before and after birth. So that's why during the fetal period, alpha to gamma 2 is the predominant hemoglobin, that is hemoglobin F, and after birth, it is the alpha to beta 2, that is hemoglobin A, that is the predominant form. Okay, so I hope this concept is clear. There is one more thing that is written here, LCR. What is this LCR? This is locus control region. Locus control region, not very important to remember for you, but you have to understand that this is the master switch. This is actually a switch. This is the master switch, because this will determine at what point of time which globin chain you are going to produce. That means during fetal period, this LCR, this locus control region, will make sure that you are producing predominant gamma globin. And after birth, this locus control region will make sure that you are producing predominant beta globin chain. Okay, so that's why this is known as the master switch. But anyway, it's not very important at this point of time to understand. So I hope it is clear. So we have beta-like globin genes. Those are epsilon, gamma, delta, beta. And alpha-like globin genes include zeta and alpha. Before birth, predominant beta-like chain is gamma. So that is why they will combine with alpha to form alpha to gamma 2, that is hemoglobin F. After birth, it is the beta. So that's why they will produce alpha to beta 2, that is hemoglobin A. Okay. Now now that you have understood this, now we will see the exact time frame at which they are getting produced. So as I told, alpha is the predominant form before and after birth. So there is no change. After around 8 weeks, the predominant, it is alpha globin chain is the predominant form of alpha-like chains. So this is predominant. As you can see here, zeta and epsilon, these are just embryonic globins. So after 8 weeks, you are not going to find this change. As you can see here, then the gamma globin will be produced. Gamma globin will be produced, and the gamma globin at around birth, the gamma globin chain is around 30%, and the beta globin is around 20% during birth. It is around 20%. So gradually the gamma globins will start to reduce and the beta globins will start to increase. At birth, alpha is 50%, gamma is 30%, and beta is 20%. Okay, so from this you can understand at birth you have 60% of hemoglobin F and around 40% of hemoglobin A. Okay, so hemoglobin A is alpha gamma. So alpha gamma will produce hemoglobin F, so that is around 60% during birth, and alpha beta is hemoglobin A, so that will be around 40%. Okay. At around 6 months of life, there will be no gamma globins. So gamma globins will be reduced to less than 1%. So at around 6 months of life, it is predominantly hemoglobin A or adult hemoglobin. And in adult life, we have very less delta globin chains also. So that will produce alpha to delta 2. Alpha to delta 2 will produce hemoglobin A2. Hemoglobin A2. Okay. Till now we have discussed all the important hemoglobins. Hemoglobin A is alpha, beta. Hemoglobin F is alpha, gamma. Hemoglobin A2 is alpha to delta 2. Okay. So adult hemoglobin, as I told, mostly it is hemoglobin A. So that will be around 95 to 97%. Hemoglobin A2 will be around 2 to 3%. And hemoglobin F is, very insignificant, that is less than 1%. So that is the composition of an adult hemoglobin. If you see the hemoglobin F, it will start around 8th week of life. At 28th week, hemoglobin F is 90%. At birth, it is 60%. It is 60%. And at 6 months, So it will continue till 6 months, and beyond 6 months, it will be less than 1%. So this concept is important, that hemoglobin F is present till 6 months. So this concept is important. We'll discuss more on this when we discuss about the sickle cell anemia and thalassemia. Okay. So I know it is, if you are studying it for the first time, it will be slightly difficult. But believe me, all these concepts are important to understand the disease better. So if you have not understood this, then there is no point in studying about the sickle cell and thalassemia, because we'll not be able to correlate the pathophysiology with the basics. Okay. So that is why I'm spending a lot of time here, so that you will be able to understand and remember it. Okay. So now if you have understood this, now we'll see. Okay, now we'll understand one more concept. This is slightly easier because you have studied it in biochemistry also. So if you see, this is the beta globin. This is the beta globin. Beta globin gene. This is the beta globin gene containing the DNA sequences for the beta globin. Now this DNA, through the process of transcription, all these things are happening inside the nucleus. This is the nucleus. So through the process of transcription, it will make the mRNA. So this is the mRNA. Now this mRNA will have a 5' and 3' and it will have exons and introns. So basically these globin genes, globin mRNAs, they will have 3 exons, 2 introns. So as you can see, exon 1, exon 2, exon 3, and we have 2 introns. So as you must be knowing that exons are the coding regions, so that is what we need. Introns are non-coding sequences, that's why introns have to be removed. Okay, so once this is formed, it is also known as heteronuclear RNA. Once this is formed, this has to be processed before the translation. It has to be processed. So those are known as the post-transcriptional modifications. There are 3 post-transcriptional modifications, as you must have studied. The first one is the 5' capping at the 5' site. That is with the help of 7-methylguanosine. With the help of 7-methylguanosine, they will be capping at the 5' site. And at the 3' site, there will be a poly-A tail. There will be a poly-A tail. This is the second phenomenon that happens in the post-transcriptional modification. Okay, so 5' capping and poly-A tail. Apart from that, what is the third phenomenon that is going to happen? The third one is the splicing. So splicing is a process by which these introns are removed, and this is possible because of the spliceosomes. So spliceosomes will come into picture and they will just remove the introns and after removing the introns, the exons will be joined together. Okay, exons will be joined together. So this is the final processed mRNA, which is having a 5-methyl cap, which is having a poly-A tail, and the introns are removed. So this is the final mRNA which is going into the cytoplasm, and in the cytoplasm, this mRNA, with the process of translation, it will form the protein or the globin chains. Okay, so this is what happens. Again, why this is important to remember or recollect this concept here, because when we will study about the types of mutations that we are going to find in beta thalassemias, again this concept will come there. Okay, so before finishing this, what are the function? Why this capping is happening? So what is the function of 5' capping? So 5' capping is actually it is helping the mRNA. It is actually protecting the mRNA from ribonucleases. So it protects from the action of ribonuclease. Also, it helps in initiation of translation. Similarly, poly-A tail, it also has 2 functions. It provides stability. And also it helps in exporting. That means export of the mRNA from the nucleus to the cytoplasm. Okay, so that is the function of poly-A tail. Okay, so we have discussed the important concepts. Now we'll just finish this with 2 hemoglobin variants. 2, I would say, beta globin gene variants. 2 beta globin gene variants. The first one is hemoglobin S. Hemoglobin S is a combination of alpha 2, beta 2S. There is some variation in the beta globin gene which will produce a different type of beta globin. Okay, so here there will be beta globin gene production, but the beta globin gene production is not normal. It will be a different type of beta globin that is known as beta S. So how this beta S is produced? If you take the beta globin gene, if you take the beta globin gene at the 6th codon, there will be a change. What is that change? The glutamine in the 6th codon will be converted to valine. So glutamine will go, valine will come. So at the 6th codon of beta globin gene, the glutamate will be replaced by valine. So this will produce the beta S or sickle beta globin. Okay, so S for sickle. So they will make this sickle beta globin, and ultimately they will produce the HbS, that is the sickled hemoglobin, HbS. Okay, now what is the property of this hemoglobin S? So actually, this hemoglobin S, whenever the hemoglobin is deoxygenated, this hemoglobin S has a tendency to polymerize. As you can see here, they will tend to polymerize, and they will polymerize in such a way that they will make the RBCs like a sickle. They'll make the RBC like a sickle, like this. I mean, they can form various other forms also. This is one of those forms. And in this process, they will make the RBCs less deformable. Also, they will increase the adhesiveness of the RBCs. I mean, there are many things that will happen when those things we'll discuss when we'll discuss about the sickle cell anemia. Okay, so this is what this hemoglobin S is going to do. Now we'll see what will happen whenever there is hemoglobin C. Hemoglobin C. This is also a variant of beta globin. In this condition, what happens? In the beta globin gene, at the exactly same site, the 6th codon, the glutamine will be converted to lysine. So if the glutamine is replaced by valine, they will make the HbS. If the glutamine is replaced by lysine, they will make the HbC, hemoglobin C. Now, what is the property of hemoglobin C? Now, hemoglobin C will activate one transporter present on the RBC membrane. That transporter is potassium chloride transporter. And once they activate this transporter, potassium from the inside the cell, it will go outside. Potassium and chloride from the inner side of the cell, it will go to outside. So potassium along with water will come outside. So that means they will make the RBCs more dehydrated. Okay, so they will make the RBCs more dehydrated. What is the functional consequences of this? That also we'll discuss later. So these are the two globin variants that you should remember. So we have discussed about the normal hemoglobins, hemoglobin A, hemoglobin A2, hemoglobin F. Embryonic hemoglobins, you can just forget about it. So three important normal hemoglobins are A, A2, and F. Now we have discussed two variants. One is hemoglobin S, another is hemoglobin C. So these are all the important hemoglobins that you have to remember. Apart from this, there are thousands of hemoglobin variants, but those are not very important at this point of time. But these 5 important ones you have to remember. Okay, so the last part is how you identify the various hemoglobins. So this is done by electrophoresis method. So this electrophoresis method is known as cellulose acetate electrophoresis at alkaline pH. Okay, so this is the cellulose acetate electrophoresis at alkaline pH. Electrophoresis you must have studied in biochemistry also. The method of protein estimation or protein determination. So basically here in this side we have the cathode, and in this side we have the anode. And we'll see the mobility of various types of hemoglobin, and from that you can identify which type of hemoglobin is present. So this is remembered with a mnemonic. You can say HAFSA2, HAFSA2. HAFSA2. So here, actually in this image, they have not shown the hemoglobin H because that is rare. Hemoglobin H, as I have already told you previously, it is nothing but the beta 4, which is found in certain alpha thalassemias. We will discuss later. So hemoglobin H will be nearer to the anode, then the hemoglobin A, then hemoglobin F, then hemoglobin S along with various other hemoglobins, not very important. Then hemoglobin A2. But here we have to understand that hemoglobin C is also present along with the hemoglobin A2. So cathode and C. So near cathode and C nearer. Okay. A and E are of at this time it's not very important. Okay, so HAFSA2, H, A, F, S, A2, and along with A2, we also have C. Now we'll see how to interpret this. If you see here, these three, this one, and this one. So all these patients will have, they are having only one band at A. So that means all of them are having predominant HbA. So that means it's a normal individual, normal. They are normal individuals. If you see here, this patient is having the band in the HbS region, So that means this patient is homozygous for hemoglobin S, HbSS. If you see these two patients, this patient and this patient, both of them have band in HbS, a slightly faint band at HbS and HbA. So that means they have combination of HbA and S. So this is HbAS. That means they are heterozygous for sickle cell. So they are sickle cell heterozygotes, sickle cell trait. Okay. Similarly, if you see here, two bands, one at HbS and one at HbC. So these are heterozygous for HbS and C. Otherwise known as the compound heterozygous for HbC and HbS. Okay. Now you must be thinking that if a band is present, for example, this, if a band is present here, how can you tell that this is C and this is not A2? Yes, from this electrophoresis, you cannot differentiate whether it is a C or A2. To differentiate that, either you can go for a electrophoresis at acidic pH, or you can go for high performance liquid chromatography. In the recent times, we generally do not use this cellulose acetate electrophoresis. I mean, I have never seen a report of cellulose acetate electrophoresis. What we use is high performance liquid chromatography. Okay. So high performance liquid chromatography will look something like this. Here you can actually see the different hemoglobins. They have, they have different retention time, and from this you can identify different hemoglobins, and also you can quantify them. So high performance liquid chromatography has the advantage of identifying and quantifying the amount of hemoglobins. Okay. So more details, I don't think it is required at this point of time. Maybe we'll discuss a little bit more when we'll discuss the individual entities. So that is all about hemoglobin basics. It's slightly difficult, so you have to see the video at least 2 to 3 times, and you have to memorize everything, so that your learning of hemoglobinopathies will be very easy. So thank you so much for listening.